Thank you for joining, and thank you to our speakers for joining this webinar today. And this is co-hosted by QuantaBio and DKSH Technology. I'm Mishna Mallinson, Business Manager at QuantaBio, and representing DKSH is Ms. Prema Tangprasithipat, Manager for Technical Sales. The talk is titled Transforming Long-Read Sequencing with Accelerated Processing Times. Before we begin, let me give a very brief introduction to QuantaBio. QuantaBio is a leading manufacturer of advanced DNA and RNA extraction and amplification reagents. We were founded in 2001 by former amplification scientists from Life Technologies and have since continued to innovate new and exciting reagents which aim to help users get reproducible results from challenging samples with simple workflows. Our approach is to combine engineered application-specific enzymes with our proprietary inhibitor-tolerant reagent composition to give superior product performance for reverse transcription, qPCR, PCR, and next-generation sequencing workflows. Today, we're really honored to be joined by three leaders in the long-read sequencing field from the pioneering Siriraj Long Read Lab, or SILOL for short, in Bangkok, Thailand. They will share some of their current work and experiences with sample processing and library preparation for medical microbiology applications. The lab is led by Dr. Tidhatit Wongsulawat, a trailblazer in long-read sequencing, and in particular on the Oxford Nanopore sequencing platform for medical research, growing and expanding this technology in Asia and globally. As well as her role at Siriraj Hospital, Dr. Tidhatit is an adjunct instructor at various institutions and a consultant for the Centers for Disease Control and Oxford Nanopore Technologies. Dr. Wolawat Kulasuwan is a postdoctoral research fellow at the Faculty of Medicine at Siriraj Hospital and is a member of the SILOL lab who is working on the integration of genetics and microbiology. And Ms. Patalapur Nimsama from the Faculty of Medicine, Siriraj Hospital, and also the SILOL lab, is a senior research associate and experimental scientist with an interest in bioinformatics. So without further delay, let me hand over to Dr. Tidhatit for the presentation. Thank you.
Thank you so much for having me here today. I come with my team, not just me. So I'm so excited that my team also share something interesting about the experience, how to use the QuantaBio enzymes and got a good result. Okay, before I move to my talk, let me disclose something that my team members and I do not receive financial support from QuantaBio or distributor DKSH in Thailand, but we come today to just sharing our experience. So first, before I move to the presentation, let me tell you something about my place. So now we are working at Siriraj Hospital. This hospital in Thailand, in Bangkok, is the largest hospital in Thailand, has 3,000 beds and outpatients per year is about 3 million. And this is under Mahidol University that support R&D as well. I came back from US to Thailand in 2021 and start a small lab called Siriraj Long Read Lab. And until now, 3.5 years, we have about 12 people. The technology that we focus are Oxford Nanopore technologies and also bioinformatics. We also work with medical field and also industrial field. And what about nanopore technology? Let me talk a little bit about this technology. Nanopore technology, here you can see the device called MinION. It's very small, it's chocolate bar size. Yeah, this is MinION. And you can see here also flow cell. Yeah, the flow cell is consumable item. When we zoom into the flow cell, we can see it's about 2048 nanopore here. And this is again zoom in. And you will see this is cross section of the nanopore embedded into the membrane. And when we turn on MinION, the device, then the sensor can detect the ionic current that pass through the pore. Once the molecule like A, T, C, or G comes to the pore, it blocks the pore, and the ionic current cannot pass through well, then the sensor can detect like this, show this kind of pattern. And you can imagine if we have like a single strand DNA pass through the nanopore, and then it can create pattern like this, and the base calling or bioinformatics software can tell us this pattern like which kind of DNA strand. Okay, and again, this is just a video to show that when we load DNA onto the flow cell, what happened. Double-strand DNA move to the nanopore and denature using the helicase, denature double-strand DNA into single-strand DNA and move to the pore at 400 bases per second. And if we have 1,000 pore, you can imagine a ton of the data came from the nanopore machine, right? And need bioinformatician to do data analysis. Okay, in Thailand, we use this machine for many things. Yeah, pathogen genomics, metagenome, virus, bacteria, fungi, parasite, for example. We use nanopore to compare with the conventional method to detect bacteria or bacterial identification, and also antimicrobial resistant gene detection. Also, we try nanopore on diagnostic field. For example, brain cancer and mitochondrial DNA sequencing. Also, we use this machine because it's portable and small. We used it in education as well to train students, give lecture, and arrange the workshop. Also, many scientists in Thailand want to use long read technology to do whole genome sequencing of many organisms, as well as synthetic biology as well. Okay. And then there are many projects that we need to use polymerase. And because of the long read, sometimes it takes some time to amplify the DNA, right? That's why we need some polymerase that can do like fast turnaround time. Yeah, long range PCR. Long range being maybe 4 kb or up to 12 or 15 kilobases, for example. High fidelity. Yeah, we need very good quality polymerase as well. And one important thing is about reasonable cost. This is just an example that why we need fast turnaround time. You know, for the preterm birth, when infection happen, and then we need maybe just same day or not above 1.5 days to get the result. This is the data to show that if we use Sanger is quite cheap, right? So if we use Sanger, but if the patient has like polymicrobial infection, Sanger cannot tell us which organism. That's why we need to use nanopore in this case. So we compare between cultivation, the gold standard, Sanger sequencing, and also nanopore sequencing. We found that nanopore can return us the result very quickly. Yeah, maybe 5 to 9 hours. But for us, we want to do better and better. So it means that we want to save more time. And the original protocol of the nanopore, even for the PCR amplification, it takes about 3 hours. So if we can save time on that, it means that we can return the result faster. Another important thing that we focus is about pharmacogenomics or PGX. It's the study of how person genome affect drug response. For example, in Thailand, the person who want to use allopurinol, this is a kind of first-line drug for gout, then they have to test that they have HLA-B5801 allele or not. If they have, they got high risk to developing the Stephen Johnson syndrome like this. And not only this allele, if people want to use carbamazepine, this is another first line drug. For Thailand, there is a guideline to say that, okay, you have to test or screen for this kind of allele before you use the drug. So then we try nanopore-based HLA typing, starting from the genomic DNA, and then we have to amplify 4 kb of HLA-B gene. So in this case, we have to use long-range PCR. Previously, it take about maybe 4 hours to do this. And then we have to use the product to do library preparation and nanopore sequencing. After that, we perform HLA typing or bioinformatics data analysis. Then we can map the data to the drug use. So you can see on the right-hand side, if we do sequencing, we can detect many kinds of the HLA-B allele and can match to the drug list on the left-hand side. That's why in this case, we try to do sequencing. And to amplify this region is quite long, right? It's 4 kb. That's why maybe year and a half ago, Quanta Bio came to us, and then we found that the enzymes that Quanta Bio offer us to try is match what we want. Fast turnaround time, can amplify long-range PCR. From the website I checked, is really long. Can go up to 40 kb or 24 kb, something like that. For us, now the longest one that we amplify in our lab is about 15 kb. Yeah. And high fidelity and also the cost in Thailand is okay for us. Not only that Quanta Bio came to us, we have to read first the application note, and we found that in principle, this enzyme maybe save us about two hours. And also I discussed with specialist Ishna from Quanta Bio as well, discuss about how to use the enzyme and also the feature of the enzyme before we make a decision. That's why today I would like to share 2 applications from my team to use this enzyme for 16S RNA gene sequencing. So now in Thailand, it's quite popular to do full-length 16S. Yeah. And I heard that the Asian microbiome now, they try to move from short read to long read as well. Another application is about pharmacogenomics will be presented by Miss Cook today. Yeah, please welcome them.
Good afternoon from Thailand, everyone. So I'm very excited to share you my experience regarding to application of the RepliQa HiFi ToughMix on full-length 16S sequencing for accurate microbial identification. So my name is Bert. I'm researcher at the Division of Medical Informatics. So today I'm going to talk about how to use this enzyme to amplify the full-length 16S RNA sequencing. So as everyone knows, that the 16S RNA or ribosomal RNA genes is universal marker gene that used for the bacterial classification. But actually, in the 16S RNA gene is contained like 9 variation regions that help us to classify at the species level. Traditional sequencing technology, for example, like Illumina, that's produce some short read that cannot allow us to capture all of the full-length 16S RNA gene. They just capture only some partial gene, for example, and V3 and V4, just approximately 500 base pair, and allow us to identify the bacterial at the genus level. However, nowadays they have like the emerging of the long-read sequencing technology. For example, Oxford nanopore technology does allow us to sequence the full length of the 16S RNA genes and give more some detail about the microbial classification at the species level. So that in this work, I'm going to apply the quantable enzyme to amplify the 16S RNA gene at the full length and sequence it using the Oxford Nanopore sequencer. So in this work, I'm going to choose the mock community because we know exactly the composition and the relative abundance of the microbial that's included in the genomics mix. In this case, I'm going to choose gut microbiome genomics mix as well as skin microbiome and vaginal microbiome genomics mix as an input DNA. So for the experiment in this work, I used like the universal primer to amplify the full-length 16S RNA gene from the mock community. And the treatment is control for treatments belong to gut, skin, vagina, and negative control. And indeed, for the reagent, we use RepliQa HiFi ToughMix. For the PCR condition, we're gonna amplify the full-length 16S RNA genes for 30 cycles and temperature at 59 degrees Celsius. And after that, we need to purify them. And finally, we got like the full-length 16S RNA amplicon that's ready to prepare the DNA library. So, and this picture show like the gel electrophoresis of the target 16S RNA gene that obtained from the gut microbiome, skin microbiome, and vaginal microbiome. And this is the concentration after cleanup with the magnetic bead. As you can see in this slide, you can find that they will show like the high concentration of the PCR amplicon up to like 220 nanogram per microliter. But in this case, I only use only 1 microgram per sample in total 12.5 microliter to prepare the DNA library following the ONT protocol. For the DNA library preparation, in this case, I use the ligation sequencing amplicon SQK NBD 114.2 version 14 for the DNA library preparation. Actually, for the ligation sequencing kit, is contained like the 3 main steps. The first one is the end repair by adding some A tail to the end of the DNA fragment. And after that, if you have several samples, you can tag with the different barcode. And finally, you have to attach of the sequencing adapter. And finally, you will got like the library of the DNA like this, and that's ready to sequence using the Oxford Nanopore sequencer. For this work, I use the MinION flow cell as a flow cell to sequence the target 16S RNA gene by using the GridION device and base calling using the super accuracy base calling model to convert the current ion to the ATCG. And this slide shows the sequencing run summary. So after running for 45 hours, this is the result that we got from the machine. We got like the base call in total 2.9 gigabase of the pass read and approximately 900 megabase for the fail read. And this graph show the blue line. That means that this is the expected read when doing nanopore sequencing, and this is the green one represent the pass read and the red one represent the fail read. And this is the least length bar graph that show the target 1.6 kb of the amplicon. And this histogram represent the barcode hit in each sample. For the barcode 13 is representing the gut microbiome, skin microbiome, vaginal, and negative control. You will see that all of the barcode hit more than 500K of the read that we obtained from the sequencer. That's enough for the downstream analysis. For the downstream analysis, after we got like some FASTQ read, in this case, we use like the web tool that's called Epitome that's provided by the Oxford Nanopore Technology Company to analyze by using the workflow that's named the FASTQ 16S. For this workflow, they use some BRAS tool to BRAS again the 16S ribosomal RNA database that's created in 2021. And this is the result of the gut microbiome. So when we're using the quantabio enzyme to amplify the 16S, especially for the gut microbiome and sequence using the nanopore sequencer, we found that most of the bacteria can detect by the 16S RNA full-length analysis, except for the Bacteroides when analyzed with the epitome websites. However, when we look at the detail by using this read to brush against with the different database, we found all of the bacteria that's labeled in the gut microbiome. As well as in the skin microbiome, they propose that they contain like 6 bacteria. And this is the result. We can amplify and identify all of the 6 bacteria inside the skin microbiome and vaginal microbiome as well. And for the processing time, as Dr. Tip said, that the quantabio enzyme help us to decrease the amplification process from like 3 hours to 45 minutes when compared with the conventional or traditional tax polymerase. And we have like 2 hours for the DNA library preparation. That means in total, we're gonna finish the experiment up to like 3 or 4 hours. And then we start sequencing and we'll get the result with the same day. And this is like my experience. And I would like to share to you guys to know about how to use this enzyme for the full-length 16S RNA sequencing for microbial identification. Thank you.
Good afternoon. My name is Patalaporn Ninsmer. I am a research assistant in Srila Longleat Lab, Faculty of Medicine, Srila Hospital, Mahidol University. Today, I will introduce about applying to comprehensive haplotyping of the HLA gene. First of all, I will introduce about human leukocyte antigen or HLA. HLA is a gene in the major compatibility complex or MHC region that located on the short arm of chromosome 6. HLA genes are relevant in pharmacogenetics. The MHC locus is divided into 3 subclasses. Class 1 consists of loci A, B, C. Class 2 consists of loci DP, DQ, and DR. And the last one is class 3, contain additional genes involved in immune and inflammatory response. HLA genes are named using specific nomenclature that provide detailed information about the gene and its variant. HLA allele name consists of up to 4 fields, each of which include 2 or more digit number and separated by colon. The first field refers to allele group. This is typically based on serological typing. The second field is related to the specific protein sequence of allele. Variation in this field can affect function of the HLA molecule. The third field indicates the synonymous mutation in DNA sequence that do not change the protein sequence. And the fourth field shows difference in the non-coding region of the DNA, such as intron or regulatory sequence. The level of detail is crucial for applications like organ transplantation, disease association, and immune response. The importance of HLA-B, first, in the pharmacogenomics, HLA-B alleles are associated with adverse drug reaction. Example, HLA-B 1502 related to the carbamazepine for the epilepsy and bipolar disorder treatment. HLA-B 1501 for the abacavir for HIV treatment, and HLA-B 5801 so allopurinol for gout and kidney stone treatment. Second, the transplantation compatibility HLA-B matching between donor and recipient is critical in organ and bone marrow transplant. Mismatch of HLA can lead to graft rejection. And the last one, HLA-B association with some disease. In routine lab, HLA-B tests were divided into two main techniques. First is serological method, and second is molecular technique. In molecular technique, the first is sequence-specific primer or SSP. SSP rely on the specificity of primer extension that is matched or mismatched with the 3' end of the template. The method is rapid identify and can be applied for low resolution HLA-B typing that report in 2 digits. Next is sequence-specific oligonucleotide probe or SSOP. SSOP rely on the locus-specific amplification comprising the polymorphic site. The resolution level depends on the number of probes. For intermediate resolution, that report in 4 digits. And the last one is sequencing-based typing or SBT. SBT involves directly reading the DNA sequence of HLA gene. The degree of resolution will depend on the length of the sequence that obtained. It is the only method with ability to detect and characterize new allele. My project focused on HLA-B whole gene amplification and long-read sequencing. This approach is faster than SBT and generates phase polymorphism with longer reads, resulting in the ultra-high resolution HLA-B typing. HLA-B typing was reported in 3 fields. For long-length PCR master mix band were selected for HLA-B amplification optimization following the recommended protocol of each band. Only one band could not amplify the 4.6 kilobase amplicon. After that, only 2 bands which were ready to use PCR master mix were selected to validate input DNA concentration and successful to amplify. However, the RepliQa HiFi ToughMix took less time compared with other bands. The result resulted in successful amplification of the whole gene and sequence to achieve ultra-high resolution HLA-B typing. So, summary, RepliQa HiFi ToughMix is ready to use master mix that easy to handle, time saving to prepare and reduce contamination. HLA-B whole gene amplification was successful with RepliQa HiFi ToughMix, providing rapid amplification speed and high yield compared to the other band. Thank you.
Thanks again to all our 3 speakers for their wonderful presentations. And really, I think this has shown how exciting long-read technology is in medical research, and I'm really sure it will grow as part of standard practice in the clinic as well, which I'm sure is where we're heading to. Before we proceed to the questions, let me just summarize with some key takeaways on the featured product, RepliQa HiFi ToughMix, and then we can go into some questions about the applications with the speakers. So RepliQa HiFi ToughMix is an endpoint PCR master mix that amplifies with extreme speed of up to 1 kilobase per second. And as we just saw, it's really ideal for long read applications, significantly reducing workflow times while maintaining that accuracy. It's a high fidelity enzyme, as Dr. Fidelity mentioned. It's 90 times versus wild type TAC and is suitable for amplifying targets up to 40 kilobases in length. It has a high tolerance for AT and GC rich regions of DNA and is suitable for NGS applications using magnetic or streptavidin coated beads as well. RepliQa HiFi ToughMix is actually also an outstanding choice for short amplicons. It was recently identified as the enzyme for choice for library amplifications for short-read Illumina and also long-read PacBio sequencing in a study from the Wellcome Sanger Institute in Cambridge, UK. In the study, RepliQa gave the best coverage uniformity and unbiased amplification of low inputs compared to over 20 enzymes, including those from companies like Kappa, Takara, Illumina, and NEB. And this is in agreement with the findings of the Stylol lab that RepliQa is a great choice for NGS workflows. So we now have time for some questions. The first one, I think, for Ms. Cook. It says, you mentioned using nanopore for HLA typing. How does this compare to short-read Illumina methods, and how does the accuracy compare?
May I help her? So actually, both Illumina, nanopore, or even PacBio or any sequencing machine can do for this gene. But for us, we use nanopore because we don't need to wait for many samples to come. Yeah, we can do just maybe 3 samples or even 1 sample. That's why we do nanopore sequencing. But if we have other technology in our lab, for example, maybe short read or something, yeah, and the price, if we do like a research, yeah, maybe 200 samples come together, then we can think again about the price of the sequencing cost. However, another good thing about the long read is about haplotype. Yeah, we get whole haplotype of the HLA. But if we use short read, we have to do like to map, yeah, an assembly for the HLA-B. So this is 2 things that we consider.
So I think this is for Dr. Bird. This is for 16S full length. How do you decide which database to use for the analysis?
So for the 16S is can map with the different database. For the long read sequencing, they have like some specific program that's that they curate that suits for the long read sequencing. But in this slide that I show like by brush again the universal database that you can easy access in the NCBI database.
Okay, great. So these are freely available databases that you can use. So from your experience with previous experiments, then you choose to use based on that, you know which database works for your samples?
So it's depend the type of the sample. Yeah. Nowadays we have like several reports that they try to curate some database that's specific to the one that you're interested in. So I think it's depend on the sample that you use.
So actually for EP2Me, when we train students, yeah, we want to show them how to throw data on cloud and then see the result quickly. But for us to do publication, yeah, it's more serious. We use different set of tools, yeah, that publish in Nature, something like that. Yeah, and also the database that we use. Yeah, sometimes we try to find the best one. Or sometimes new publication just came. Yeah, and then we want to try that one as well. Yeah.
That makes sense. Yeah. For the whole team, maybe it says, what is the optimization like? Does it take a long time to optimize the protocol?
For the 16S, actually, if we use nanopore based kit, I mean now they have 16S available. Yeah, just for you to purchase. Yeah, but that primer cannot capture bifidobacterium. That's why for us, we have to find some primer that can capture important bacteria. Yeah, for gut microbiome, for example. Yeah. And that one is take about 2 months, right, Dr. Bird? Yeah, to do optimization. But in some cases, we use 16S from nanopore to do pathogen detection because we don't care about the bifidobacterium. Yeah. So it depends. That's one, no need to do optimization. Yeah, just
So,
follow the protocol said. Mhm.
okay, great. Yeah, and that's actually, I see the next question. Can this work with Oxford Nanopore 16S barcoding? So you answered that. So you have also tried 16S barcoding kit, but it depends which bacteria you're targeting. Do you have a protocol for 16S barcoding with Illumina sequencing? Or maybe I can answer that one. So yeah, I mean, as Dr. Tidatip and her lab have explained previously, the advantages of using the ONT workflow for speed and for full length 16S analysis. We do also have from QuantaBio and for other publications, we have some protocols for barcoding with Illumina sequencing using RepliQa HiFi ToughMix as well, if that's your preferred method. But I think today we've seen both are valid sequencing methods. You see last one here, it says, thank you for the nice presentations. Do you have any explanation why one species in the gut microbiome could not be detected?
Based on like my experiment that I said that I amplify the full length and browse again the 16S RNA database, right? And the database is curated in 2021. As it might be, it's not suitable to detect all of the bacteria discovered in the gut microbiome. And the other one is the primer that we use is like some universal primer. It might be Larry cannot capture all of the bacteria in the gut microbiome. And the other reason that I think is depend on like the composition of this bacteria in the genomics mix that I purchased from the company. If the DNA content is very low, it's hardly to capture, even we amplify them as well.
Did you make a comparison between different polymerases? How did the sequence quality and the mock composition differ? Was RepliQa comparable to other polymerases or better or?
So actually, we did not compare the result yet, but for the processing time, we tried to compare the processing time.
Okay, so that was your main comparison there, the processing time. Yeah. But the data, as you've shown, was good with RepliQa and was able to give you answers. Thank you for the nice presentations, Ms. Cook. Can you comment on how ultra-high resolution sequencing compares to standard PCR methods used for HLA-B in terms of cost and turnaround time in a clinical context? So how compares to PCR for cost and time?
In the turnaround time, the RepliQa is faster than other company. So the sequence can be reported in at least 1 and 2 days.
And if you compare sequencing to old standard PCR methods, how does the cost compare?
The cost of the sequence is higher than the conventional PCR, but we get the high resolution, the HLA-B typing for the screen, the adverse drug reaction and other such as the transplantation or something associated with the HLA-B.
Great. Yeah.
And we previously tried other enzymes. Yeah. And it took us about 4 hours.
Oh,
yeah.
wow.
Mm-hmm.
So it's quite a big difference in turnaround time with the enzyme.
Yeah. So it's about the management in my lab. If we have to keep PCR run overnight, this is maybe not good. Yeah. So if we can just finish everything within 1 day, this one that I prefer. Yeah.
Do you see, as you move forward, other places in the workflow you might find some improvement, like the extraction, DNA extraction, or the analysis time? Are these also places you're working to improve the speed?
Of course, yes. We consider from the beginning to the end as well. How easy to extract, and minimum number of the items that we need to use, something like that. Yeah, because we think not just only our lab. We try to transfer the technology to other labs as well. Yeah, that's why we think from the beginning to the end. Even develop bioinformatic part to do 16S data analysis and HLA-B typing as well.
Fantastic, full workflow. Thank you very much to our participants, and a huge thank you to our excellent speakers. If you have any additional questions, you could contact me. My email address is there. Or Dr. Thirsip, sorry, I've pasted that wrong. That should be a link to the long read lab, but you can Google long read lab and you'll find the contact details for the lab. So please contact us or contact directly for more information. Thank you very much.
Thank you so much, everyone.
Thank you. Take care.